up at the top of the hour here. So first of all, there's a state science and technology plan that's being developed that is done in accordance with the EBSCOR program that's led out of UK, but this is a real opportunity for the University of Louisville to engage more broadly with the state around science and technology and the kind of interaction between science and technology and innovation-based businesses. So just thinking about some of the talks we're going to hear today, I mean, the first talk related to machine learning and medicine, I think that it's easy for us to think about how clearly the work that we do connects to industry and competitive industry in the state, but the state not all the actors in the state always realize that. So this science and technology plan for the state is an opportunity to make that more apparent. There's a company called RTI, which is out of RTI region, who is the lead on that. And so some folks have already spoken to RTI. We'll be convening groups of faculty for them going forward. So in the coming weeks, probably there'll be some opportunities for folks to be involved in. I think they'll be doing more like group types of interviews for faculty coming up. So just a heads up on that. The second thing is that with President Schatzel is really interested in seeing a research strategic plan be developed at UofL, which is great. It's great timing. So we've started pulling the data pieces of that together, where, what's the data that will help drive decision making around a strategic plan, and then we'll have engagement, a lot of opportunities for engagement around constructing a strategic plan that will, in some places, be disciplinary based. So there'll be contributions from individual colleges and research centers and things like that. And then, more a larger framing of overall university strategic plan that might, to make sure that we capture interdisciplinary types of research initiatives. So that's pretty exciting. It's something that she recognizes is needed, and it's a great opportunity for us to convey the importance of research and a strong research mission and what we need to put that, to advance that. A lot of people on the call here, a lot of our active researchers have engaged with our representation in Washington, D.C., and I'll just say that it's the appropriation season. We've been working really closely with our congressional delegation on advancing a number of really exciting initiatives on behalf of UofL faculty and researchers. So we expect that, I don't know what you can expect out of Washington, D.C., so I hate to say I expect anything at all, honestly, but we're as well positioned as anybody is to make the best from what can be made out of the current situation in Washington. For example, the Chips and Science Act, a lot of people are familiar with. There's a lot of opportunities that are coming down the pike because of the Chips and Science Act. New regulations among them, but in addition to new regulations that we have to meet, there are opportunities. For example, UofL was engaged in 3 different NSF engines. This is NSF's version of kind of a technology-based economic development, with use-inspired research at the heart of it. We led one. We were involved in 2. It looks really promising, and that we're going to be getting all 3 of those. So looks really good. We don't have official word yet, but it looks really positive. Also, we have been for about a year trying to position ourselves for these regional technology hubs that will be coming out of Economic Development Administration. So we went together with University of Kentucky about a year ago now to visit Washington, D.C. and meet with our legislators and try to get language introduced into the bills that would be favorable to our state and our institutions, and we were successful in doing that. So we expect the regional technology hubs first call for proposals for the I forget what they call it, the planning grant, or the first phase of those to be coming out in the next couple of weeks. And so we're in a strong position to respond to that. And we already have plans for topic areas, and we'll be going in together with University of Kentucky as a lead, and probably institutions from Tennessee as well. So a lot of action going on there. So I think that's what I've got for updates. So let's turn it over to the real reason that we're here, to hear about some research highlights. And to start off, Dr. Huang and AI and machine learning. So
thank you so much, Kevin. Thank you so much, Kevin, for the opportunity. So I'll share my screen. All right. And everybody see my screen okay?
Right. We see your whole screen right now, your entire desktop. There we go.
That looks good. Okay. All right. So today I want to talk to you about AI in perioperative medicine. So when we talk about perioperative medicine, we mean that before surgery, during surgery, and after surgery. I'll get into this just a little bit. Those are my disclosures. So perioperative mortality is the third leading cause of death. So for people who die around surgery time is pretty significant. And this is just after heart disease and cancer. So there's a great opportunity for us to do better. Can anesthesiologists help to reduce? And we lead the care for patients coming into surgery, really from the decision to have surgery until they discharge. Now we're really getting into the long-term recovery for those patients too. So how can AI be used in perioperative medicine? We really have three goals for surgical patients. One, we want to identify at-risk patients, which patients are high risk, which ones are low risk. And second one is we want to detect the complications early. When you do surgery, there's always complications, and we want to know that early when treated them timely and effectively. So right now, AI has been used in perioperative care, ICU, pain management, drug delivery, and drug discovery. So this is really my working environment. So as you can see, as the anesthesiologist doing cardiac surgery, and I have a lot of monitors. So this monitor tell me about the electrocardiogram, the heart rate, the blood pressure, the pressure in the heart, the oxygenation level. This tell me the breathing about the patient, what's the pressure in the airway, and how much is the patient breathing. This tell me about the cardiac output. This is my EMR, electrical medical record. And this is the echo machine. So I know all the information about the heart from this. And this is a smart pump. So this can give medication on those pumps. And then I have a camera. I can see what the surgeon do. As you can see from this picture, there are a lot of information coming to me while work in the OR. So how do I combine those information together to make a reasonable judgment on those cases are difficult. And you're only human. So sometimes we'll get distracted. Sometimes important information will not be delivered to me. So that's where AI and machine learning will come in handy to help us handle those complex cases. So when we talk about AI, there are really multiple terminology. So AI is the big circle. Then inside that, there's machine learning. And then inside the machine learning, there's deep learning. So meaning we try to extract higher level information on those data. Then we'll talk about the natural language processing. So how can AI machine learning understand the language? And we'll get into that just a little bit later. So there are different types of AI. There's supervised learning models, such as logistic regression, decision trees. So we will have an expert like myself to label the data, and then the system will learn. Then there's an unsupervised learning model, so like PCA, K-means. So they only look at the patterns and association of the data. They do not rely on experts to mark those data. And more in medicine, healthcare, we're using more reinforced learning. So we'll develop a model, then the model will learn from its own actions. Is this achieving the goal, or this not achieving the goal? And there's a reward and a penalty system. Then there's more and more talk about deep learning, use multiple layers of connected neural networks, just like the human brain, so that can extract, give us higher level features from the raw data. And the connection can be backward or forward, and it's just like what we do in the human brain. And there are different weights and biases on those information exchange too. And natural language processing is something we use to try to understand spoken and written contents. And for any physician, any anesthesiologist, we have a very complex, structured, and sometimes unstructured data source for us. We have the EMR with the population health data by the state or the federal government. We have a lab system, we have a radiology system. We have the monitors, the arterial line, the pressure in the heart, the echo. And then we have wearable sensors that actually being used more and more nowadays. We have the pharmacy system, we have the billing system, we have a quality reporting system, and we even have a research and education system. And in the operating room, we have a separate OR management system. So how do we use data is very complex. And I'm going to give you some examples regarding how AI is being used. So the first example is unsupervised learning. So what they did was they have all the medications, they all have labels, they have warnings, adverse effects, and therapeutic effects. So they just put all the data together, use machine learning, and figure it out which medication will have certain therapeutic effects and which medication have adverse effects. So this is pretty significant, and because there's, especially when there's a new medication on the market, we really don't know what kind of issue we'll have with those. So this is use unsupervised learning model. And this model uses supervised machine learning. They analyzed 13,000 patients going through surgical procedures. About 9% had low blood pressure during surgery. And so this machine this system uses 70% to train the model with different machine learning models, and 30% test the model, see if it works. And they were able to achieve a positive predictive value of 96%. And in anesthesia, a lot of things we do are closed loop. I give a medication, I want to see how it works, and then I'm going to come back to see maybe I made the wrong decision. Maybe I should give less, or give more. Use reinforced learning, as we talked about before. So the system can learn by its own actions. So if this is working, then they will reward the system. If this is not working, will penalize the system. So they can develop a system actually to control the blood pressure and also how deep you are asleep. This is the brain electrical activity, and they were able to get pretty good results on that. And sometimes the decision I make in the operating room are very complex and very difficult to precisely use a mathematical model to analyze those. So they developed a system called fuzzy logic. So instead to give an accurate number, give the heart rate number, they will define that as mild, moderate, or severe. So it's blood pressure, pulse variation, and this system was able to make a substantial degree of accuracy to tell me how sleep the patient are, or how hypovolemia the patient are, how dehydrated the patient is. And this system uses decision tree-based learning to predict patient analgesia, so how much pain medication they need. And the system we use clinically is I give a patient button, so if they are hurting, they can press the button and give them medication. And so this system was able to use decision tree-based learning to predict which patient will need more, how adjust those. Then there's a neural network and deep learning. And sometimes we have so much information clinically, such as the electrical activity of the brain, the blood pressure, the heart rate variation. It is very difficult for me to analyze all the data, to select the features and get a conclusion. So with the deep neural network, it was able to analyze all the hidden layers and give me the output that I want. And then we talk about the natural language processing. Why that important? As a physician, we write notes all the time. We leave a note in the EMR. Not only I want to tell others what are the important findings, but also I want to tell them what's my thinking, what's my thought process, how did I drive to this conclusion, and it can help to identify surgical candidates, assess adverse events, and facilitate billing. And this example showed that you can actually have a very high sensitivity, and although with a lower specificity to predict which patient had adverse events on that. And we're using a whole lot of images in medicine. And we use ultrasound, we use CTs, we use X-rays, we use MRI. It's a over time, I've been doing this for 21 years. I was able to identify structures very easily, but for new learners, it's very difficult. So can we use AI machine learning? So this is actually epidural catheter placement that we do this for pregnant lady, so they don't have pain in labor. So this is the ultrasound picture. So use AI machine learning, we were able to identify the epidural space very accurately. It's only off by 1 millimeter, and that's actually very good. And the depth we also use AI machine learning to predict the depth of anesthesia. One of the major jobs as an anesthesiologist to make sure you sleep. And so this study actually showed it's pretty accurate. We can predict if you sleep or not. And this one used a neurofuzzy system to predict and got a 92% accuracy. And this system is more interesting. So they say, how about we do something that can do what the anesthesia does? So they use automated system. So they call that make sleepy, but check how sleep the patient are. Is the patient having any pain? Is the patient moving? They were able to achieve the function of the anesthesia 70% of the time. It's getting close. And this system actually used to when the patient off the mechanical ventilation, and it works pretty well. The other major issue I have when I do anesthesia was low blood pressure. And all the studies showed if you have low blood pressure, you're not going to do well long term. And there are multiple studies to how to predict low blood pressure. I'm going to go to this study. I thought this study is very interesting. So if you look at this study, so they try to predict is the patient's blood pressure going to be low after surgery? And what they found is this is the clinician, the blue line. So our prediction is okay. I mean, we do all right. And we about the every end of the curve is a 0.657. And if I add the machine learning model together, I get a 0.725. But you use the machine learning alone, actually you do better, got 82%. And some of the issue we have is the information that AI gave are not clear. I don't know how they got that conclusion. So this system actually tell me what are the factors they consider. I'm just going to do through the time. So we use a lot of ultrasound to do a lot of procedures in anesthesia, and the AI machine learning can help me predict. It can tell me which is a nerve, which blood vessel, which are muscles, which are bones. And so this is the new research we've been doing with University of California, San Diego. So they were able to develop a wearable ultrasound. So instead of using the big machine you saw in my picture in the OR, this one is a tiny little patch. I can get all the picture of the heart, and they use AI machine learning was able to give me easily usable information about the patient. And the other challenge I have in the OR is I don't know how long the surgery will be. And this system actually used to predict exactly how long the surgery is going to be. They consider certain experiences, anesthesiologist experiences. And we talk about natural language processing. We have several experts in the audience, more expert than me. So they actually, in anesthesia, we're able to use this to detect heart failure early and to figure it out. We all try to do standardized protocol, but every person is different. So this system can help me to understand the subtle differences so I can do better on those. So what are the limitations and the implications of AI in medicine? The first thing is the transparency and the trust. So a lot of the AI machine learning algorithms are black box. So they got a conclusion for me, but I really don't know how they got the conclusion. That's why we need explainable AI and provide me with a reasonable clinical explanation and accountability. And what they showed with AI machine learning is root association, not a causal relationship. And that can be an issue for me to make a decision. And if the training to the training set they use are underrepresented by certain population, then if I apply this in the underrepresented population, this definitely will give me a bias. And then there's a lack generalizability. So a system developed by Harvard might not be used for UofL Health. So these are some examples that we use. I will not get into it. So I think the future directions for AI machine learning in preoperative medicine, the first one is, as anesthesiologist, I can be trained to trust this. But how about the patient? If the patient come in and want to have surgery, have anesthesia, I tell them I'm going to use the machine to help you, and will the patient trust that? And how do we help with that? The other one is I'm a physician. I am not a data scientist. I am not an engineer. I need to partner with data scientists and engineer. So I definitely welcome collaborations for everybody on the call or everybody who is not on the call. And the data that you train the algorithm must be valid and representative. And the interpretation of the data must be clinically meaningful. And as anesthesia, we should partner with the other providers, surgeons, intensivists, nurses. And AI can be used as a new tool to continue innovation. So in conclusion, AI has the potential to impact the clinical practice of anesthesia, and they have different ranges, and it's crucial for the clinicians to provide clinical practice-based insight to assist the development of AI. So we have to be at the table. Otherwise, our job might be taken away by them. And so I have a lot of collaborators, Dr. Bad, Dr. Chen, Dr. Lan, and also would like to thank the clinical trial unit at UofL, the UofL Research Innovation Office, UofL RRB, and UofL Health Research Office. Thank you.
Thanks, Dr. Juan. That was great. Super interesting. I can see the utility of it from your very first picture of the operating theater and what that looks like. It's pretty amazing. I guess it's not all just Chat GPT. There's other uses for AI and machine learning. Good to know.
Definitely.
Okay, so if folks do have questions, just throw them in the chat. Otherwise, let's move on. Dr. Mohammed to talk about some more miracles. Magic.
Yeah.
It's all magic to me.
Yeah, thanks so much for the invite. You guys can see my screen now, right?
Yeah, it's
That's good.
yeah, there you go. Yep.
Yep. Okay, so I'm just gonna talk today about the cardiac tissue slices to model cardiac health and disease in culture. So I want to say that it's not only me, we are a team between bioengineering and the Department of Medicine. We have Dr. Guru and Ayman from bioengineering, which we've been collaborating on this project for the past 5 years or so. So what's the problem? The problem basically is that the first time any drug touches the heart is in clinical in phase 1 clinical trial in humans. So this is the first time any drug, cancer drug or any disease, any drug during development. Usually, all the preclinical testing is being done in mouse and rat and in silico and in vitro and so on. But the first time any drug touches the human heart is in the phase 1 clinical trial, and that's why many of the drug fails in this during this phase because they could be cardiotoxic. And cardiotoxicity basically was one of the top tier of toxicity in any drugs. So what we did, we made a simple, reproducible, medium throughput culture system to fully preserve the human heart slices, viability and functionality for at least 6 days. So this time is enough to test any drug toxicity at least the subacute drug toxicity. So how we did this, we get usually a human donor hearts from the transplantation network or big hearts. We can do it in pigs, we can do it in rats, dogs, any other animal bigger than the mouse. So the mouse is too small to slice, but anything big has heart bigger than the mouse, we can do it. And then we cut the left ventricle into blocks like this. We put it on a vibrating microtome. So we cut 300 micron slices, and then we culture it under electrical stimulation, oxygenation, and we have optimized the media as well. And this keeps them in culture to do for 6 days to do any assessment that you would like to do. So what could you get as assessment? So you can add your drug or you can add your gene therapy to these heart slices, and then you can see the impact on calcium transient, the impact on contractile function. As you can see here, you can hang the slice in an organ path and you can measure the contractility, and you can isolate adult cardiomyocytes from them and assess the electrophysiology on them. You could do metabolic flux, you could do transcriptomics, electron microscopy, immunofluorescence, and we collaborated with a group in Colorado as well to assess the single myofibril contractile function as well in these slices. So why did we ever get into this? So basically, I'm not a heart slice person generally. So I'm someone who's interested in developing gene therapies to treat heart failure and to replace the loss of the myocardium in the heart after a heart attack. And I used to do 2 approaches, either reprogramming approaches or induction of cell cycle. And this reprogramming approaches is now is being developed by Tenaya Therapeutics, where I was the first scientist there. I spent there for 2 years, and then I went back to academia. And now they are doing the phase 1 clinical trial with this therapeutic. But and basically what we did, we generated this human heart slice technology to test these gene therapies in human heart. As you can see here, These are 4 cell cycle factors that induce cell cycle in cardiomyocytes, and you can see this phosphorestone H3 is a marker for the cell cycle induction, and you can see here we can see this induction of the cell cycle in this group only, not in the control group. So, but once we published this technology, I got a phone call from the FDA, from Alex Ribeiro on the FDA, and he said, you know, we are really dying for a technology to test cardiotoxicity reliably on human heart tissue. Are you interested in testing this? So I've never done cardiotoxicity testing before. So we collaborated with Igor Efimov and Javed Moslahi in Tennessee. And we started to validate this system for cardiotoxicity testing. So we choose 3 compounds. These 3 compounds, 2 of them, the doxorubicin and trastuzumab, they show toxicity on monolayer culture that the FDA is usually using, but the sunitinib are not showing cardiotoxicity. So we tested these 3 on the heart slices, and the conclusion of this study, and we published this study 2 years ago. So the conclusion of this study was that the heart slice culture system could detect all the 3 cardiotoxicity of these 3 compounds. But not it's much better than, so which is much better than the monolayer approach of human iPS-derived cardiomyocytes. So this actually took us to become a part of a bigger consortium for cardiac safety assessment, which is called the HESI Cardiac Safety Committee. HESI stands for Health Institute of Science, Health Environmental Science Institute. And this Cardiac Safety Committee basically is, the aim of this is like a clinical trial on cardiotoxicity. So this committee has 38 sites worldwide between pharma companies and FDA and the European regulatory body, the Japanese regulatory body for drugs, and other labs around Europe, Canada, US, and Japan. And the aim is to have a unified platforms that can test for cardiotoxicity and validate these platforms. So each site out of these 38 sites is having a platform to test cardiotoxicity, and each site received 12 cardiotoxins blindly. So the cardiotoxins were blinded, and there was 1 negative control and 2 positive controls as well added to this cohort of compounds. And the conclusion of this study was that at least in our hands, the tissue slices detected all the 12 out of 12 compounds in cardiotoxicity. The human iPS cardiomyocytes, which is the monolayer system that's been in use for a while, detected only 4 out of these 12 compounds. So overall, I think this demonstrates how reliable these heart slices in detecting cardiotoxicity. The consortium is now gathering a big publication together. So this is basically, we are still working on this. But I think what's more exciting is we took this even further to emulate all the cardiac cycle in the heart. So we can basically emulate all the physiology and cardiac physiology in culture. So to do this with the collaboration with Ayman and Guru in bioengineering, and with the brilliant bioengineering students that we had, we have developed this system where we can emulate all the 4 phases of the cardiac cycle. So this bioengineered system has got a pneumatic driver driving air in and out here, which pushes this silicone membrane, which pushes the cardiac slice up and down during the contraction and relaxation phases. And this is a graphite electrodes in the cover to electrically stimulate them as well. So basically, we are providing all the movement and all the electrical stimulation that you can get in the heart. And with this, So you can see here, this is the hot slices in culture, and you can see here they are actually moving up and down during the contraction, and we can fire electrical stimulation during the phase, the end of this of the relaxation phase. So with this, I think with this system now, we can emulate the mechanical stimulation as well as the electrical stimulation during the culture system. And with this, we reach viability for 12 days. So we extended the viability even up to 2 weeks, almost 2 weeks, basically. And with this, we could emulate all the pressures inside the culture system. So we can even emulate the disease conditions. We have some disease condition like hypertrophy and overload, pressure overload. So we can induce this in this new culture system. And this is something that we are working on at the moment. And at the end, I would like to highlight, that this technology has now put us a lot of collaborations around the world. Basically, not only here, we have collaborators in UK, we have collaborators in Germany, we have collaborators here. We have a lot of collaboration with industry as well, like Merck and Tenaya Therapeutics. And basically, we recently licensed this technology to Anabis because we've been dumped with too many requests to test, from companies and from labs to test for their targets on these hot slices. So we tried to unload our lab work and license this technology out. So we just licensed this to Anabis, which is a CRO in San Diego, that they test for cardiotoxicity. And I would like really to thank my group. These guys are really great. They work day and night, and we get human hearts. We can get it at midnight, and they stay all the night processing them and so on. At the end, I would like to thank you for the opportunity to present our work, and I'm happy to take questions.
Thank you, Tomer. That was really super interesting. If folks have questions, just throw them in the chat. But let's go ahead and move on. So a little bit more about the heart, I think, from Alex to talk about e-cigarettes and cardiac arrhythmias. Ready, Alex?
Yes, I'm just loading in. Are you seeing my screen?
Yep.
Okay,
Looks
great. So full disclosure, I was told to have 2 to 3 slides prepared for today. So expect a much briefer talk. This work has been performed in collaboration with and as part of the AHA Tobacco Regulation and Addiction Center with Dr. Aruni Bhatnagar and Dr. Daniel Conklin. I will be highlighting mostly the in vivo effects of e-cigarettes, but the work is also done in collaboration with Matt Nestoriak, who is looking at the in vitro effects in human-inducible pluripotent stem cell cardiomyocytes of e-cigarette flavorant chemicals. But the data I'm going to share with you today is primarily focused on the effects in vivo in mice and the role of ingredients in e-cigarettes in e-cigarette-induced arrhythmias. So you may be aware that the prevalence of e-cigarette use has skyrocketed over the past decade or so, and this is especially true for youth, in whom we've seen increases of up to about 20% prevalence in high school age children and about 5% prevalence of use in middle school age children. And this is concerning because we just presently don't know the long-term impacts of e-cigarette use, nor do we have the best grasp on the short-term impacts of use. Nonetheless, it's presumed that e-cigarettes are a safer alternative to smoking by many, but this is still, in several respects, unproven. And it's unclear what the impacts of e-cigarettes are on the heart specifically. First off, just some basics on e-cigarettes. If you're not already aware, there are multiple generations of e-cigarettes. To the left is the Cigalike or first generation e-cigarette that was followed by the vape pen that allowed sort of the do-it-yourself edition of e-liquids and produces thicker clouds, so higher particle counts and potentially higher levels of toxins. In addition, following that was the mod, or the sort of vape or e-cigarette with modifiable settings. So you can adjust the voltage or the temperature of operation. The tanks are also larger. There are more wicks, so this produces even thicker clouds. So a higher, more rapid dose of nicotine often, and production of additional constituents that may cause harm. And then the fourth generation pod devices, including Juul, are sort of more recent. They overtook the market in about 2018, 2019, and they've been recently followed by the disposable pod devices. But among all of these different kinds of electronic cigarettes, they universally contain solvent vehicles, propylene glycol and vegetable glycerin, as well as pretty much every time flavors and nicotine. And I'm not going to be addressing cannabis products in this talk, nor in my studies presently. But even heating and thermal degradation of vegetable glycerin and propylene glycol, which e-cigarettes do to generate aerosols, produces aldehydes, which are chief among the harmful and potentially harmful constituents generated by e-cigarettes. As well, reactive oxygen species are produced from heating these solvents. In addition, nicotine and flavors, the heating of them produce additional breakdown constituents as well. And the impacts of flavors and nicotine inhalation are unclear in many respects. So I'm going to divert real quick with some background on the endpoints that I look at and will be focusing on right now. And really just today, for the sake of brevity, I'm focusing on cardiac arrhythmias. Specifically premature ventricular contractions, which is generally what they're called in the clinic. Experimentally, they're often referred to as ventricular premature beats. So these are called PVCs or VPBs. And small increases in the frequency of these arrhythmias, which are shown on the left with the red arrows, are associated with increases in cardiac mortality. So on the right is data from a study looking at a patient population. These are individuals that were referred to a Holter monitoring clinic for ECG evaluation. And those who had more than 12 premature ventricular contractions a day had significantly increased likelihood of cardiac death, a threefold higher likelihood of heart failure related death, a sixfold higher likelihood of sudden cardiac arrest mortality. So as few as one of these ventricular premature beats every 2 hours in humans can predict adverse outcomes. The underlying hypothesis of the work in my lab has primarily been focused on the role of the autonomic nervous system in e-cigarette and aerosol-induced cardiac pathophysiology. I've also branched out into investigating the role of the individual chemical constituents. So it could be the flavorants or the solvents themselves or nicotine. But shown here on the left is just a sort of model individual inhaling e-cigarette aerosols. And upon arrival, those aerosols, we know, trigger irritant reflexes that then evoke autonomic nervous system imbalance. This imbalance is characterized by, at least in mice, a parasympathetic or rest and digest dominance that is immediate, and then is followed by a sympathetic fight or flight reaction that accelerates heart rate. And we determine this and monitor it by using implantable ECG radiotelemetry. So we collect the electrocardiograms real time during exposure to these e-cigarette aerosols. We evaluate them using advanced software. We assess ECG morphology. I'm not going to get into that today. We do also assess heart rate variability and heart rate in terms of changes from air controls. So we recently published our findings that the menthol e-cigarette aerosol produced by blue menthol increases ventricular premature beats in mice when they are exposed acutely. This exposure only involved 3 9-minute puff sessions with 2 puffs every minute. So that's a total of 54 puffs. So just very short-term exposures can induce arrhythmias in mice. Interestingly, the tobacco flavor that is produced by blue did not induce ventricular arrhythmias. And we also found that propylene glycol solvent aerosol alone significantly increased these arrhythmias, but the vegetable glycerin did not. So we followed this up by looking at the impacts of short-term exposure to Joule aerosols. And here also we found that the menthol flavor, but not the Virginia tobacco flavor, increased heart rate and decreased heart rate variability while also increasing these ventricular arrhythmias, suggesting that menthol is uniquely arrhythmogenic. In a separate study, we evaluated the impacts of the solvent aerosols with nicotine added to them using nicotine salts, which are now found in Juul and sort of the more modern e-cigarettes. And when we examined mod aerosols, we found that there was sort of a threshold level of nicotine that was responsible for the arrhythmogenic effects of these e-cigarettes. In addition, we incorporated a drug test, a drug-induced arrhythmia test using isobutanol, and found that nicotine underlying e-cigarette induced susceptibility to ventricular arrhythmias. This shown here is an instance of ventricular tachycardia in a mouse that was exposed for 3 days to nicotine-containing e-cigarette aerosol. And then more recently, we've looked at synthetic coolants, which are present in the ever increasingly popular ice-flavored e-liquids. So these are kind of taking the youth vape preference by storm. And if you look at convenience store shelves, usually about half to a quarter of the available flavors have ice in their name. So within these e-cigarettes is a synthetic coolant, either WS3 or WS23 or both. And we found that by adding these synthetic coolants at commercially relevant levels, the e-cigarette aerosols of these mixtures did evoke the arrhythmias, whereas the vehicle with the nicotine and the solvents did not. And the effects corresponded with increases in heart rate and decreases in heart rate variability, suggesting sympathetic dominance. So we evaluated the correlation in heart rate and also heart rate variability, which is not shown here, and the instance of these arrhythmias, and found that only with the coolant aerosols did heart rate elevations positively correlate with ventricular premature beat arrhythmias, suggesting a sympathetic mediation of this arrhythmogenic effect. So with that, I thank you for your attention. I'd certainly like to thank my mentors and colleagues, Matt Nestoriak, Dan Conklin, and Aruni Bhatnagar, and I welcome any questions you may have.
Thanks, Dr. Carl. Super interesting. I just have one real quick question, and that is whether, so there's all this work on e-cigarettes and the relationship with these arrhythmias. Is there a body of work and knowledge already around old-fashioned cigarettes and these types of cardiac issues?
So one thing I did not mention is we evaluated the impacts of conventional combustible cigarette smoke in that same study. And while we tried to adjust the exposures to have equivalent nicotine levels delivered, we did see that these arrhythmias were not induced by the combustible cigarette aerosols. Mm-hmm. However, or rather smoke. However, it's well established that smoking increases the likelihood of sudden cardiac arrest. Mm-hmm. And the immediate induction acutely of cigarette smoke I'm not sure how well established that is, but I will say in this case, this is the first published study to demonstrate that e-cigarette aerosols cause spontaneous ventricular arrhythmias.
Yeah, yeah, that's great. Thanks so much. Interesting, sobering. All right, couple of quick announcements. So Lee Botner is going to talk about the NIH All of Us research program.
Thanks, Kevin, and thanks for each of the presenters. I appreciate everyone sticking to time, even though some of you had more slides than were recommended. I appreciate it. Yes, the NIH All of Us, I believe is what it's called, is a program that I highly encourage everyone to take a look at. Basically, the program is building a data set to help researchers expand health data, particularly from diverse populations and those that are underrepresented in biomedical research. The benefits for researchers is simply that you would have access to various different cloud-based and centralized and secure data sets from various different institutions and settings, and from researchers at all different career levels that include students and early stage investigators. And it includes data such as survey responses, physical measurements, electronic health records, genotyping arrays, whole genome sequences, Fitbit records, biospecimens, and all kinds of different things. And there's various different tiered membership levels. There is some information on a website that has all kinds of great links. There's a lot of different resources there that you can tap into. They often have some various different webinars about it, and it is something that is slowly getting built, so it will become more robust over time. And it is a good resource for building your proposals. So I just wanted everyone to be aware that it's something new.
Thanks.
And if
Thanks,
anyone
Lee.
has questions, you can reach out to me.
Thank you, Lee. Okay, Bailey.
Hi, everyone. I'll be very brief and hopefully give you a few minutes of your afternoon back. I just wanted to invite all of you to come celebrate innovation and entrepreneurship at U of L with us on April 18th. We are having our annual, our quasi-annual, our regular Entrepreneurship and Innovation Awards, celebrating anyone who's worked with industry or gotten a patent, IP, licenses and options. We really want to celebrate the commercialized research that's happening at U of L. As you know, our other event that we do fairly regularly to recognize your work celebrates research and scholarship. This is innovation and entrepreneurship, and I would love to see all of you there. Kevin and our new president will be, they will be handing out several major awards. So come celebrate with us. I'm going to drop the registration link in the chat and hope to see you guys there.
All right. Thanks, Bailey. I look forward to that. It's always good to recognize and celebrate the amazing research and translation that comes from our community. All right, that's it for today. Thank you again to the speakers. Those were great presentations, and I will echo Lee, and you guys did a great job keeping the time. So that was nice. I didn't have to do any reminding. So appreciate that very much. All right, thanks everybody.
Yep, thank you.